Let me sort of remind ourselves that influenza, despite having a good vaccine, still can cause a lot of problems. And if you look at this, one of the most recent statistics from WHO, we have HIV with 1 million, but flu is really up there with 500,000 deaths, and even hepatitis C is quite high. Now, in terms of the United States, those are the latest CDC numbers, and again, over the last 6 years, we have between 12,000 and 56,000 deaths in the US alone, and a numerous number of hospitalizations, and the number of people who have been infected per year. These are annual data by estimates, 9 to 60 million people. So one of the reasons we do have this problem, and not everyone can be vaccinated, is that we have a lot of variation in terms of influenza viruses. And this shows the different subtypes we have. We have 18 influenza A virus subtypes, and they are divided into 2 groups. And the blue one here includes the H1 and the H2, which are human subtypes for the hemagglutinin, and then the H3. So we have circulating over the last 100 years H1, H2 in the blue, group 1, and H3 in group 2, and influenza B viruses are over there in terms of the hemagglutinin. And then you should also look here at the scale, which is 7%. So if you look at the distance of these different hemagglutinins, we are talking about 30–40% difference in the different hemagglutinins, and that's why you can understand that it is very difficult to have one specific vaccine which would take care of all of these different influenza viruses. And our goal is really to make a vaccine which would protect us against all strains of influenza A and B viruses, and hopefully induce a long-lasting immunity. So let me also remind ourselves that in the US we have 3 different kinds of influenza vaccines FDA approved, and that is the inactivated, usually formaldehyde inactivated vaccines, and the live attenuated one, which are cold-adapted influenza viruses, and then more recently recombinant DNA, sorry, too much, David, DNA, recombinant proteins, and those 3 have all been FDA approved. Now, the complexity of the influenza virus vaccine can be easily seen in terms of what are the components at the present time. And at the present time we have, so this is a 2016-2017, and so we have an H1 component, we have an H3 component, and we have 2 different influenza B virus components. And over the last 15 years, only 2 years did not have any change in terms of these components from the previous year. So that means basically every year new vaccine preparations have to be made, manufactured, and that makes it quite difficult. The other point is that if we do have a pandemic, usually a new hemagglutinin subtype appearing, it is very difficult to develop and to make vaccines. And in the last pandemic, which we experienced in 2009, we were basically too late. And I think many of you have seen this curve here. The blue curve is when the vaccine in 2009 was actually delivered, and the maximum activity in the US was where the red curve is, and this really represents October, and here we are into December. So this is when the manufacturer was able to deliver the vaccine, and then obviously it takes a long time to get to all the Walmarts and CVS's and pediatric offices, and then it takes another 10 days until the vaccine really works. So we were much too late in terms of the pandemic in 2009, even though the government really did spend a lot of money, and the manufacturers really tried to do their best. And then also another point is that even though the vaccines are really underutilized, they are underappreciated, the vaccines sometimes, or at least some components of the vaccine, are not optimally matched, and then we have up to 20% of all the emergency room visits in the US, particularly on the East Coast, are associated with influenza virus infections. So all of this makes it very difficult. Now, again, let me stress that influenza virus vaccines are unique, and they have to be given annually because, as I said, there is a lot of antigenic change. Now, I want to sort of talk a little bit about this antigenic change and contrast it with what is going on with measles virus. So measles virus is also a negative strand RNA virus, like influenza, and the mutation frequency of measles virus is actually at least in the same ballpark, but it's very, very similar to that of influenza viruses. In other words, the RNA-dependent RNA polymerase of measles virus is very, very similar to that of flu. And so we ask ourselves, why is it that measles virus has not changed, at least not over the last 60 years? Because we are using in the current vaccine the same vaccine strain which was developed, which was used when the vaccine was developed in the 60s. Okay, so what we have tried to understand is there something which allows us to understand why measles is sort of not changing, static, and flu constantly. There's a constant antigenic drift, and obviously a consequence of this antigenic drift, also antigenic shift. And we have done this by trying to understand the flexibility of the influenza virus hemagglutinin versus and also the neoaminides versus that of the glycoproteins of measles virus. This was done mostly by a postdoctoral fellow, Nick Heaton, in the lab, who is now a faculty position at Duke University, Ben Fulton, who is a PhD student in my lab, and Ben Hurley, who is a faculty member at Mount Sinai. So what we have done is basically, let me sort of give you a brief description of the experiment we have done. Is we took the measles virus genome and the influenza virus genome and inserted, and this is the slide for the influenza virus hemagglutinin, and we inserted by transposon mutagenesis 15 nucleotides, that is 5 amino acids, basically in every single position of the genome of influenza viruses. And what we can see here on the left is the one trimer of the hemagglutinin with its head and with its stalk. And so again, we introduced at every single position, 60–70%, a 5 amino acid insertion, 5 amino acids, 15 nucleotides, and then ask the question, how many of these insertion mutants are viable? And what we can see here is the green is part of the stalk, and this is part of the stalk, and then the red is the globular head. And then after we rescued all of these insertion mutants, asked the question, which part of the influenza virus genome is most tolerant towards these insertions, we found something very interesting, and this is after the rescue. So these are 100% of all the mutants, and if you look here, these blue, whatever, 12%, is in this position. It's in the globular head. The light blue as well, yellow, orange, this yellow, this blue is here. Even this orange, if you go all the way up, is still in the globular head. And if you go to this one here, the blue one, it is already outside of the coding sequence. So this suggests that the majority of mutants which are viable, and that the domain of the influenza virus, as you will see, is really the most mutable one, the one which is most flexible in allowing the insertion of these 5 amino acids, is actually the globular head of the influenza virus hemagglutinin. In contrast, when we do the same thing with measles virus, which doesn't have a segmented genome, for flu we had to do every single gene separately, we had every single RNA segment. For measles virus we had to do it in one shot. So again here we have the different gene or genes coding for the proteins of the measles virus, and important are here the surface glycoproteins H and F. And we did basically the same experiment, inserting insertion mutants, insertion of 5 amino acid insertions, and then we asked on the what can be rescued. And if you look down here, you probably can't read this, but this is the F and the H. So basically, the F and the H of the measles virus are not tolerant towards insertions. And one can sort of look at this in terms of a comparison of all the influenza A viruses. We have here the hemagglutinin, and the hemagglutinin, this is the total one. If I would just use the head, it would be even much higher, the tolerance. So these are arbitrary units here, and we can't really compare measles and influenza directly, so we have it normalized to 1 here, but you can see here the F and the H doesn't allow any insertion. So we believe that the hemagglutinin of the measles virus is has functional constraints, and has to do that the receptors are proteins. It has to interact with many different receptors on the cell surface, and that this is the reason that the measles virus hemagglutinin in the virus is actually much more stable than flu. And I have shown that the head of the hemagglutinin, which is the immunodominant area of the virus, really tolerates quite a lot. So this is really, I think, interesting to understand why one virus is more stable as compared to the other. Nevertheless, what can we do about a better influenza virus vaccine? And we believe we can make a better influenza virus vaccine. And this is a collaboration of 3 laboratories at Sinai: Florian Kramer, Adolfo Garcia Sostres, and my own. And in essence, what we are trying to do is we are trying to make a vaccine where we present the conserved domains of the virus as a vaccine. So what we should understand is that we have all been, everyone here in this room has been infected with the influenza virus. And if we talk about the H1, which is one of the components of the influenza viruses which are circulating right now, we all have seen some of these viruses, and so we have some antibodies against the much more conserved stock, but most of the antibodies which we have, most of the immune responses are against the head. The problem with it is that the virus constantly changes, so that an antibody against 2017 influenza virus will not be protecting us if you get infected in 2019 or 2020. So what we are trying to do now is to vaccinate with a chimeric hemagglutinin, which has, in this case, we have what we call is a chimeric H5, which has a head of an influenza virus, which we have not seen as humans, and we take the 2017 stock, and then, so this will be the first dose of the vaccine, and then we follow it up with the second one, where we have again the stock of the 2017, but now we have yet another hemagglutinin head. So basically what we are trying is to so since we have not seen the head here, we are naive against this head of the H5 and H6 in this case, and we are hoping that the immune system in humans will be redirected towards the much more conserved stalk, as well as the neuraminidase. So these both are, the stalk is immunosubdominant, as well as the neuraminidase is immunologically subdominant as compared to the head. But by using these irrelevant heads, we hope that that this will induce an immune response which can be protective. Now, mice are not humans. We understand that mice are not men, and also, as you will see, ferrets are not humans, but it works beautifully in mice. Now, mice are all naive, so we have to actually give them some dose here. We have to prime them, and we prime them with a DNA, in this case here, a chimeric H9. And what we want to do here, we want to show that by immunizing against the stalk of an H1, we get complete protection. So what we are trying to do is now we have here CH6 and CH5, but we can use any other H18, etc., in terms of making these chimeric structures. In the mouse, we have to use protein not to get confused with what the NP might do, etc. So in this case, we are just giving the DNA expressing the H9/1, chimeric hemagglutinin 9, and the stalk of 2017, and then we boost with proteins, and then we look what happens. So we get some immune response against the head and against the stalk. If we then now vaccinate with a different head, we get again some head antibodies, more stalk antibodies, and yet more stalk antibodies as we vaccinate with a second dose. And then we ask the question, can we now protect the mice which we are vaccinated that way? And so in this case, for example, we used the 1934 stalk, and we protected against the 2009 hemagglutinin, the Netherlands. So this here is, on the left side, we have the weight curve for mice which were vaccinated, which are the purple here, and dark purple, dark blue here is the vaccinated one, and we get basically very little weight loss, which suggests that the mice are very protected. And if one just looks at the survival, in both cases, the control and the stalk vaccinated animals are actually completely protected. Now, we also have done this in terms of different subtypes, so basically, as long as we have a stalk belonging to group 1, all of the mice are fully protected. In this case, we have used H5 challenge and H6 and have vaccinated them again with the stalk vaccine. So this works very, very well. It also works very well in ferrets. It also works very well in terms of guinea pigs. So we have quite a lot of data in terms of animals, but again, mice are not men, etc. What have we done in terms of the mechanism? The mechanism is clearly antibody mediated, because we can take the sera from mice which we immunized the way I described before and just passively transfer them into naive mice, and we get, again, complete protection, as can be shown here, and that goes also not only against all variants within one subtype, but also from one subtype in group 1 to another subtype in group 1. We have also data in terms of the group 2, so those are these hemagglutinins up here, and again, it's the same procedure that we use a group 2 stalk and put different hemagglutinins heads onto it. And I should stress so that in terms of having a truly universal influenza virus vaccine, which would protect against all strains, not only in one subtype, but against all subtypes, we would have to have 3 components. We would have to have a group 1 stalk, a group 2 stalk, and a influenza B virus stalk. So that's quite important in terms of that vaccine approach. We are agnostic about what kind of platform should be used, whether it's an inactivated one, whether it's a live virus platform, whether it is a protein platform, or whether it is a David Wiener DNA platform, because in our case it depends really what we provide as an immunogen. So we are very interested in the mechanism of these antibodies. How do they work? Because they are not hemagglutination inhibiting antibodies like the ones we usually get when we vaccinate against the head. So the antibodies against the head prevent the virus from attachment. The antibodies against the stalk do not prevent attachment, and the mechanism by which these broadly protective stalk antibodies facilitate neutralization is quite interesting. And the way we are doing this is we have again this influenza virus mouse model, and we give the antibody IP 2 hours before we challenge these viruses intranasally, and then we check weight and morbidity, mortality in terms of all the way out to 2 weeks. And what it turns out is that the kind of antibodies which we generate by the vaccination which we are using is actually an ADCC-dependent mechanism. It is one, and this is work together with Jeff Ravitch. We are able to show that these broadly neutralizing hemagglutinin stalk-specific antibodies require Fc receptors. And we have shown this on the left side here, where we look at the blue curve, which is protection with antibody against the stalk in normal mice. And then the red curve here shows these are mice which are lacking Fc receptors, and they are not protected at all. And on the right side, we have the survival curves. So this was quite interesting, and we are trying to work more on trying to understand this antibody-dependent cell-mediated cytotoxicity, which is probably the major effect of these stalk-specific antibodies, and that is that we have an infected cell or a virus, and then the Fc portion of the antibody is actually interacting with the effector cell. And in humans, it's the FcR3, and in mice, is Fc receptor FcR4. Now, okay, let me give you the bottom line right away. We found that these antibodies work by a very interesting mechanism, namely that there are now 2 interactions with these effector cells, not only through the Fc portion of the antibody and the Fc receptor, but there's also sialic acid involved. And that is shown here. So if we have a hemagglutination inhibiting antibody, it sits up here, prevents attachment of the hemagglutinin to another cell which it gets infected. And the effector function here of the Fc and the Fc receptor here is shown as on the left side here. On the right side, we can see that actually the effector cell we have actually 2 sites of interaction. We have the interaction with the Fc of the stalk-specific antibodies and the Fc receptor, plus it turns out the sialic acid on the effector cell, which interacts with the hemagglutinin binding site. And this is very briefly, once more, we do find ADCC only with the antibodies on the receptor side, on the stalk side, but not on the head of the hemagglutinin. And we were able to show that these antibodies really need, in order to get the best ADCC activity, we have to actually have this sialic acid interaction. We were able to block the sialic acid interaction with a FAB fragment, and also by mutating the receptor binding site. And let me just show here on the left side, we have, this is the ADCC signal, but if we have an FAB fragment which protects the sialic acid binding site, we actually eliminate this, and we can also eliminate this ADCC activity by changing the hemagglutinin binding site with a single mutation. So I see this is, the clock is running. Let me sort of repeat, summarize what I have said. The mechanism by which these stalk-specific antibodies are working and are optimally operational is that there are 2 interactions, one through the Fc portion of the antibody and the Fc receptor, but there's also a sialic acid binding of the hemagglutinin to the effector of the sialic acid binding site with the sialic acid on the effector cell. So we hope that we will be able to develop a universal influenza virus vaccine by reducing the immunodominance of the hemagglutinin head and increase the immunogenicity of the hemagglutinin stalk and the neuraminidase. And I have talked about the optimal activation of the Fc-mediated effector functions, and that this is done by a two-point contact, and that this is what really is making these antibodies very effective. And let me just acknowledgement again, this is a collaboration of Florian Kramer, Dr. Fias Austria, my own lab, Jeff Ravitch, in terms of mostly the Fc receptors, Patrick Wilson, I haven't really talked much about that collaboration, and Sriram Subramaniam, who does the structure work for these chimeric hemagglutinin molecules. And we have a very talented group of students and postdoctoral fellows who have pushed forward this program over the last several years. And we are supported by NIH, PATH, GSK, and the Gates Foundation. Thank you very much.